are. Now we are. Okay, great. Fabulous. Ready to rock and roll? I heard some pings.
Yeah, good. It was
shiny. Okay. So on Friday, we had just started our tour of the cell. Remember, specifically in this case, we're looking at the eukaryotic cell, which is larger and somewhat more complex. It's also the cell that you and I have. Also tends to carry a larger variety of organelles, specifically that have membranes on them. So we had started inside out. No, that's rude. We had started at the nucleus, right at the very center, which is also the information center. Okay, and remember, keeping in mind that the DNA, camera's inconvenient for me. So our nucleus has DNA in it. Okay, and that stores all of the information for everything in the cell. Okay. So remember, we wanted to think about our nucleus as this giant library inside of which are all of these books for all of the information that we would possibly need. The nuclear envelope is like the walls of the library that keeps all of this information safe. You'd think I'd get used to having this thing on and stop stopping it every couple of seconds, but it's fine. Okay, but the DNA are like the books. Books are not going to leave, so what actually leaves our nucleus? The RNA, good. Okay, so RNA is what's actually going to leave that nucleus, right? We want to leave that DNA intact. Okay, we never want to mess with the source code. Okay, just like when you go to a library, you're going to copy stuff down. Okay, but you never change the original book. Okay, you're going to take notes. Those notes go with you, and that's what you do stuff with. Okay, so we left off then, just barely starting to talk about, okay, now we have this information. Woo! Now what do we do with it? Right? Do that. I'm going to leave my mini roadmap running across the top to kind of keep track of where we're going. All right, so let's make us something. Okay? So we're going to find that there's lots of different things we can make. The first thing we're going to try to make is proteins. Why? Because it's our favorite. So if we're going to go make a protein, we're going to go to a ribosome. Okay? Now remember, ribosomes are one of our few organelles that don't actually have a membrane. So ribosomes are found in both eukaryotic and prokaryotic cells. But proteins are so important that that makes sense. So if we want to make a protein, am I still on there? Yeah, just barely. If we want to go to make a protein, we're going to go to a ribosome. Okay, so let's talk about ribosomes for a hot second. Okay. So there are two kinds of proteins. So we're going to look at the second side here. This is where we're going to focus. Both of which will make some kind of, there are two kinds of ribosomes, both of which are going to make some kinds of protein. Okay, so if you're going to take one thing away from here, ribosome, something, something protein. Okay, that's the big key. Okay. We can find proteins in two places. The first of which, okay, the cytosol, which is another word for cytoplasm. I mean the same thing, right? That free-floating gel of an ocean. Okay. So these are called free ribosomes. Right? And these are the ones that you kind of saw for the most part in those images. They're literally loose and on their own, living their best life, not tethered down by anybody. The other place that we see ribosomes are on a form of endoplasmic reticulum. Man, I didn't name these things. You're just going to have to go on the ride. Okay, we can shorten that to ER. Okay. In this case, okay, the ribosomes stick to the endoplasmic reticulum and make it look kind of bumpy. Okay, because ribosomes kind of look like little round balls when you're looking at the cell. Okay, even when we blow them up, they're not the most spectacular looking of organelles, right? Not that we judge. So for our endoplasmic reticulum, when we have ribosomes attached to it, then it becomes the rough, because it looks bumpy. ER, right? Now, regardless, if our RNA goes to either of these places, right, something with a ribosome, doesn't matter, we're going to make a protein, because that's what ribosomes do best. They make proteins. Okay, now as you might imagine, what kind of protein you end up with is going to differ probably, but we don't care about that. Okay, right now, keep it sweet, keep it simple, something protein. I want to focus on what is the job of a ribosome, and no matter where we are in the cell, we're going to make a protein. That feel okay? We'll keep our track at the top, so we're going to do some erasing. Okay, so let's look at the endoplasmic reticulum itself. So we already know about this left-hand guy, right? That's the one that has ribosomes on it. So if our RNA is going to the rough ER, okay, we already know the main thing it's going to kick out is something proteins. Right, all sorts of stuff with proteins in it. Okay. One of our goals is to what can I focus on? I only need to remember one or two things. Proteins is still our big thing. Okay. If we compare that to the smooth ER, remember our rough ER is rough. If we look at our photo down here, we have the two types. We can see on the rough ER, which is on the right side of the photo, all of the little ribosomes on there, the little dots. And all these little dots make our rough ER look bumpy. Whereas on the left-hand side of the photo, we have the smooth ER. Okay, so we still have, you can see the membrane on both sides, so it's a membrane-bound organelle. But our smooth ER is all membrane, no bumps, right? So our smooth ER, which is lacking ribosomes, okay, still makes primarily biomolecules. This looks a lot familiar, doesn't it? Well, that's not how you spell biomolecules at all, is it? Let's try that again. Bio. There we go. We're still making biomolecules, but we're pretty making everything but proteins now. Okay, so I'm making lipids, I'm making carbohydrates, okay, all of the other ones that we like, and talked about in our last unit, well, we can't make a protein. I can't make a protein because they don't have a ribosome. Okay? Ribosomes are specialists. It's the only place largely that you can make proteins. You can do all sorts of cool things with those proteins, but you have to have the ribosome there to do it. So if we need to make another biomolecule, then we're going to go to the smoothie R for that. Okay, so our endoplasmic reticulums really divide and conquer the making of those biomolecules, right? Our rough ER makes the proteins. Our smooth ER pretty much makes everything else. And that's how I would remember that. And that's also how I would focus on that, because those are their major jobs. They do a bunch of other small things. Okay, but at their core, that's largely what they're doing. Okay, still feel good? Okay, so my RNA has a choice. It either wants to go to a ribosome to make a protein, or it's going to go to the smoothie R to make something like a lipid or a carb. Got to pick one or the other, right? We can't go to both. Ooh, let's see all this stuff here. See if I can click around a little more, huh? Okay, so at this point now we've made something. Okay, so if I made a protein, I would ship, ooh, whistling a little. I would ship it, can't stop. Ship this around the cell to somewhere where it is needed. Okay, now what if, okay, any questions about the making of things? You feel shiny? Okay. What if I had something, I had some junk food, and I needed to break it? Okay, what do I do to get rid of stuff? Okay, or I made too much, and now I need to get rid of it. Okay, what would be my process for doing that? Okay, so that's what we're going to do when we look at breaking things. Just started more to the left. All right. Okay, one of the main ways we break things down, and I think the only one we're really going to look at here, is called a lysosome. Okay, and this one's not too bad. So there's going to be a couple of new terms, but there's one key piece here that's going to feel very new. I'm really sick of this yellow. So lyso, okay, looks like the word lice, which we've seen before, right? Remember hydrolysis? Okay, so we've played with this term before. Okay, so we know that the word lyse means to break. Because remember when we did hydrolysis, we broke down, right, large compounds into smaller compounds. In that case, specifically using water. Okay, and that's the key here. So this is a sac, okay, made of our membrane. okay, specifically our phospholipid membrane. That's a B. Very dramatic B. Okay. All of the sacs in here, okay, we'll find with our transport proteins, we'll find with our lysosomes, are pretty much all going to be made out of our phospholipid bilayer, right? Because it wants to come out. If it comes in, it needs to be coming in from the plasma membrane. If we need to get rid of this stuff, it also needs to be able to go out the plasma membrane. So this stuff needs to be able to touch the edge. So it all needs to be made out of the same stuff. Okay. Inside our sac are hydrolytic enzymes. Okay, now here's a bunch of words that we've never put together before, but we know all the pieces, right? So we've done hydrolysis. This just means to break using water. That's not new, okay? We've literally done that, right? We've drawn that out. That's not too bad. Okay, we did that so much we were kind of like huffing at it. We were so sick of it, right? Okay. Enzymes, okay, we've seen before. What kind of biomolecule is an enzyme? Protein, our favorite, right? So we know that we're using fancy proteins to help break down chunky things we want to get rid of, or at least make smaller using water. That's what that sentence says. We have a special sac to do it. Okay, so this is basically like the stomach of the cell. Write me autocorrect. Okay, so that's literally what's happening here. We're taking large molecules, okay, and we're going to break them down, okay? Many of which we've all seen before. Proteins, lipids, right? Large carbohydrates, that's what a polysaccharide is, right? Remember, saccharide is the core word for sugar. So we're going to take large chunks of them and make them smaller chunks. Okay, maybe in that form, that's the form that the cell wants them, and they might be used. Maybe they're going to get thrown away. We're making cell poop. Okay. In most cases, and this is a key feature, your cells are very sustainable. Okay, waste not, want not. Okay, so a lot of this stuff does not get trashed. So if we break down a large polysaccharide into small carbs, those carbs are very likely to be recycled into something else. If we break down a large nucleic acid into small bases, those bases will be recycled and be made into new RNA and DNA strands. Okay? Very little of it gets trashed completely unless it is absolutely not needed, it is damaged beyond repair, or there's something wrong with it. It's like a toxin or it's a bacteria or stuff like that. Okay. So more often these things are taken in, if we've taken in like a cheeseburger sized thing on the cellular level, we'll take something that size and we'll break it down into usable bits and then push those around into areas we can use it. So we've taken a large polysaccharide, broken it down into smaller monosaccharides, and we'll move those around into areas where we can use the small parts. Okay, so the lysosome, the stomach of the cell. Very similar job, very similar type functioning, right? Your stomach uses enzymes and acids as well. This is using just simple enzymes to do the job. Taking big things, breaking them into small things. Feel good? Okay, so now no matter what we have, if we've made something like a protein, or if we've broken something down into smaller bits and we have these smaller bits, okay, now we have all this stuff. Okay? And these things just can't leave, say, the rough endoplasmic reticulum or leave the lysosome and walk around on top of that gushy ocean of a cytoplasm. So it sort of begs the question of how exactly are things moving around and how do they know where to go inside the cell? So I just got them saying, well, when we break things down in the lysosome, then the cell can use those parts. Well, how do we know what to use and where it needs to go in the cell, right? So that all happens because we have a very efficient shipping center. Okay, we're gonna move our map at the top. Okay, so the Golgi apparatus also, you hear me lazily call it, but many other things do, called the Golgi Body. Okay, both are correct. Serves as this function. Okay. The Golgi Body is made up of membranes, right? Stacked membranes together. Okay. And these stacked membranes are called cisternae. Okay, so I like to think of them as like partially filled Ziploc bags. They're all kind of stacked on top of each other. Okay. So these stacked cisternae, or stacked Ziploc bags, make up like the Amazon warehouse, or the FedEx warehouse. Okay, and this is what's making that shipping and receiving center. I can actually draw in white, so let's get color. All right, so if we look at the image in blue that we have in our right side of the screen, this is our Amazon shipping center. Okay, so we can see these sort of folded sacks on top of each other, and these are the cisternae. Okay, so this is a unidirectional warehouse. So if we had just left, say, the endoplasmic reticulum, just made that protein. And now I need to figure out, well, where does the cell need this? I made it for a reason, but we need to send it somewhere. Okay. So things move around the cell in what's called a transport vesicle. We see that word here too. Okay, just like our lysosome, okay, our transport vesicles are also made. Is that green showing up okay? I'm sorry. Are also made from that phospholipid bilayer, aka they look the same as our plasma membrane. Put a little pink protein in there. It's very technical looking. Okay, so I'm obviously drawing it as like one thin little line, but we're going to imagine with that phospholipid head and the little tails tucked into each other and another phospholipid head. Just like our membrane will look. Okay, so our transport vesicles otherwise act like little cars or boats if we're still imagining the cytoplasm being an ocean. All right, so we've left the rough endoplasmic reticulum and we're going to head to our Golgi body because the Golgi body, like an Amazon warehouse, knows who needs what in the cell. Okay, so I'm going to head towards my Golgi body and I'm going to head towards what's called the cis face. Okay, which if we imagine this Golgi body as stacked bags of Ziploc bags, okay, this is the humped or curved edge. Remember I said this is unidirectional. Okay, cis just means, right, same. So everyone's going in the same side. Okay, so we're going to all enter in that curved edge. All right, so this is the receiving end of our warehouse. Okay, at this point, the transport vesicle is going to open up, drop our protein off onto our warehouse, and it's the Golgi body's problem now. Okay, but that's okay because this is what the Golgi body does, right? Okay, so if it needs to be modified, okay, membranes need to be added, caps or tails need to be added, we need to polish it up, whatever, okay, that can happen, but otherwise it does exactly what you think a shipping warehouse would do. It's going to take a look at it, okay, it has all of the receptor inputs from the rest of the cell, it's going to sort it out, put a little label on it, package it up in a new transport vesicle, okay, and ship it out. I'm really whistling bad today. Ship it out the opposite end, what's called the transface. Just meaning opposite or other end. And this is going to be, from this case, the curved edge. Okay. So again, you're getting this unidirectional motion, right? Everybody comes in one side and leaves the other side. Okay? It's like a conveyor belt, a giant Amazon warehouse conveyor belt. Everybody comes in one side, gets sort, labeled, stamped, and is shipped out the other side. Okay? And once we're shipped out the trans end in a brand new transport vesicle, there's my very accurate looking protein on the inside. Okay? We have a new shipping label on us, so we know where in the cell we need to go, wherever that protein is needed. So a little better than USPS does lately. Oh, that was pretty good, right? Okay, everybody understand the job of the Golgi body? Okay, so usually at one point or another, pretty much everything that's made or broken down is going to come here, right? Because the Golgi knows things. Okay, so here we are. We've come a long way. Okay, so remember, we started with that was probably better, there we go. DNA here in the nucleus. In order to leave, we needed to make RNA. Okay, then if we wanted to make a protein, We went to one of these ribosomes. You can see them pretty clearly on here, all these sort of like yellowy dot things. If we want to make a lipid or a carb, we're going to come out to the smooth ER. Okay. Once we make something like that, we're going to be in our little transport vesicle, hit our Golgi body. Okay. In this image, it's a lot bigger, so you can really see the cis side, right, that nice curved edge. Right, packaged and shipped, and come out the trans edge. Okay, into a new transport vesicle. Okay. On this edge here, now yellow is not working for me, you can't see that. There we go. We can see the importance of having our transport vesicles and our lysosomes, okay, being of the same material as our plasma membrane. Okay. So if I want something to leave my cell, okay, and I wouldn't make something just to ship it out of the cell, that would be sort of silly. But if I did, okay, my transport vesicle would need to, okay, open up out of the plasma membrane, right, like a little door, so I can dump it out. Okay. For that to happen, I would need to fuse with my plasma membrane. Okay, otherwise, I'm going to end up with a weird hole in my plasma membrane. I have to like punch a hole, open it up with like a knife. Go, little friends. Okay. We don't want holes in our plasma membrane. Okay, remember the whole point of the plasma membrane is it's creating like a barrier of protection. So when my transport vesicles bind with my plasma membrane, we want this to be very seamless. Okay, and the only way that's going to work is when something like a transport vesicle binds with it, if it's the same stuff. So it's basically just adding its material in and then just sort of like vomiting whatever it has inside out. Okay, so using images like this are a really good way to practice, like we just did. I started with DNA, went to RNA, went to the smooth. Okay, practice the journey that we've created. And we've got a few more things to talk about, but these are a lot of the major large organelles that have big functions. This is a great way to practice. Any questions about the things we've talked about so far? Say that again. Now, what do you mean by parts of the diagrams? So for lab, yes, like you guys saw, but for lecture, for the most part, it's understanding the pathway. So I'm more interested that you understand what each organelle does and how they have a role with each other. All right, so if I'm here, where would I go next and why?
Okay, so somebody in the chat asked, when talking about the journey, do you mention the cytoplasm or do you just say that it travels from the rough ER to the Golgi?
So it's a good question. So the question is asking about the cytoplasm. So the cytoplasm itself, right, is not an organelle. So one, as long as you understand when you are in the nucleus, when you're not in the nucleus. Otherwise, the cytoplasm doesn't matter unless you're specifically making a big deal about free ribosomes versus rough ER ribosomes, right? Which we didn't make a huge deal about what the difference between them was, right? Good questions. Anything else?
Are all of the circles with the little dots in them, are they all like transport vesicles?
Correct. Those are all transport vesicles. And in this case, they're all just different options. All right. So at any point in the cell, there's more than one transport vesicle going on.
somebody asked, is this chapter on the test Friday?
Yes. Anything we get through before Friday is on the test. Good?
Mm-hmm.
Okay. All right, let's try a mentee. All right, you guys know the drill now. Stop it. Mentee.com. All right, and here's your code. Okay, remember, try to do this without looking at your notes. Okay, these make really good practice test questions.
It says to the art.
Yes, it does. Okay, what does the rough ER do? Makes proteins. What does the Golgi Body do? Okay, those were easy. Everybody hated those. Let's look at your next less popular option. Okay, so if you didn't choose lysosome, what does the lysosome do? Okay? So our smooth ER, okay, is the correct answer. Majority of you got that, so great job. So the major job of the smooth ER is to make lipids and carbs. So great job. You guys were unfettered despite how many things we talked about today. Good job. Okay. So there are a few more things that we need to talk about that do not necessarily go in any particular order, but are still needed in order for the cell to be alive and functioning. Okay, the first of which is to create some sort of energy. Okay, so run away. There's three major organelles that do this. Okay, the first two we're going to look at in more detail, and this third one we are not, because it's a little more rare in literal frequency in a cell. Okay, but the first two you probably recognize. Okay, first of which, of course, is mitochondria, and membrane-bound organelle. The powerhouse of the cell. Okay, so we'll end up talking about this a lot. We're also going to talk about cellular respiration in general in class. Okay, so literally the ability to break down food, something like the sugar, and turn it into ATP. Okay, ATP, adenosine triphosphate. Okay, literally cell currency. There's pretty much only two forms for the most part of currency that a cell can use to make things happen. Okay, the fuel that we use, for example, to make those transport vesicles move around, like your cars use gas. Transport vesicles also need to use something to move around. Okay, ATP is it. Okay, makes most things in fact happen in cells. So being able to make it, big deal. Okay, chloroplasts are also another big one. Okay, plant exclusive, of course. And these, of course, are using sunlight as a major process to create that same ATP, right, this cellular currency. Used for the same processes, just how you're getting to that point is a little different. Okay, so we're going to talk about those first two in a little more detail in a hot second. So instead, I'm going to take a minute here to talk about peroxisomes. Okay. So peroxisomes, again, we can break down this word and get a lot out of it. Okay, so at the end of this, okay, we can see that we're using some sort of enzyme breakdown. The front of this looks familiar though, right? And it sounds exactly like what it is. Okay, we're going to use hydrogen peroxide. That hydrogen peroxide, the kind that you pour over cups, makes the bubbling. The bubbling that you see is a form of oxidation. It's a high energy process. Okay. The byproduct of that is water. It seems to be the byproduct of a lot of the stuff we do. Okay. When you're done, okay, all that energy, that bubbling that we gave off, okay, there's our currency again. Okay. These are very similar to lysosomes, okay, in that they're little sacs that can be found in cells. But compared to chloroplasts and mitochondria, they're much smaller in number, and the amount of ATP they make is much less. Okay, but they do exist, and they are making ATP. Okay, they're just doing it this very specialized way. Not so bad with me so far? Okay, so let's take a look at our mitochondria in more detail. Okay, mitochondria, as I mentioned, is a membrane-bound organelle. All of those membranes are called cristae. Okay, the fact that it has so many membranes are really important, as we'll find. Okay, remember, the goal of this is to make ATP. That's that currency. Right. And this comes down to a big key. If we look down here, we see that there's a lot of zigzaggy membranes. That's that cristae. Okay. Having all those zigzaggy membranes is like having a lot of countertop surface area in your kitchen. The more countertop you have, the more cooking you can do, the more cookies you can make, right? Okay. The same thing here. Okay. The more countertops we have, the more cristae we have, the more ATP we can make. Okay, and we know that ATP is important. Okay, so the fact that we have all of these cristae, this extra folding, makes more room, okay, more surface area, for that ATP to be made, which is why the mitochondria is the powerhouse of the cell. It's the most efficient, or one of the most efficient organelles at doing this because of all of these folds. All right, you guys know the drill. Okay, we will pick up and talk about chloroplast and then a little bit about the cytoskeleton on Wednesday, and then start talking about diffusion, which is the cell transport stuff, which I will probably slim down a little bit. Don't forget to do the Socrative, of course, our teacher's question. And otherwise, happy Monday, happy studying.